So I'm going to record this and we'll put this on, I have a channel on YouTube. You actually can go in and take a look at this. I'll put this on right now as we go along. So the origin of species, chapter 24. We talked about Chucky Darwin and going to the Galapagos Islands. He found stuff there there's nowhere else on Earth. Just like if you went to Australia, there's things there that you've never seen before. Because it didn't evolve here. It's only there. They had a show the other day on Australia. Pretty cool place. But there's more deadly spiders and snakes there than there are in all the United States, all North America. Pretty wild place. You know the story about mice in Australia, right? Did I tell you about that? So there's a lot of things that we've done that have kind of changed things. What's this? Looks like a dodo bird.
I was going to say Colorado or something. It's got deformed wings.
Yep. So, you know, some birds and species have become extinct. This one here, if you look, see it's got some oil on it, right? So it kind of looks like a dodo bird, but it's more like it's an ocean bird. And it's just gotten, look at the rocks, they're all covered with oil and messed up. We've changed our environment a great deal. Does anybody remember the Exxon Valdez? The Valdez? No. There's an Alaska, big tanker up there, ran aground, and there's mess everywhere. One of the first ones that they ever came upon. Now, you know, with the pipeline that's coming down through this way, everybody's up in arms about that. And we could have a spill somewhere, and it could cause a lot of damage. But who knows what's going to happen with it.
Yeah, we just had one.
What's that?
We just had one in Northern Ireland.
Spill? They're there. They just keep them quiet.
Yeah.
Yeah. And at some point, somebody's going to blow one up. You know what's going to happen. It's a terrorist, or you know. Anyway. So speciation is the origin of a new species. So are we still creating new species? Sure. Unfortunately, we're losing probably more species than we're creating. That's not a good thing. We are on the road right now to a catastrophic problem in the world. We are killing lots and lots of organisms off. We have no clue we're even killing off. There are organisms out there we haven't discovered yet that we are killing off, that we're becoming extinct, that may be helping to balance out our world. So we are at that point in evolution that we have to be careful. We could lose a lot of the animals on this earth just because of the pollution, the acid rain, the carbon monoxide, carbon dioxide being produced that we are producing ourselves. So we have two types. We have microevolution. These are adaptations that occur within a population. And we have the macro, which is the evolutionary change above the species level. So we're looking at the whole ecosystem is changing. I tried to watch Biodome this weekend, but it's not on Netflix or Amazon, so I could not get it. That's a funny flick if you ever watch it. So I watched Son-in-Law instead, which is equally stupid, but entertaining to say the least. So species is a Latin word for kind or appearance. The same kind of appearance, so they look alike. Now when you look at mosquitoes and flies and insects in general, you have to look very, very closely to see the different species. You have to look at their wings. Their bodies may be identical, but their wings may have one little partition that's different in it. That's the only difference in the species. It makes them special. So we look at the morphology. We look at what the size, the shape, what they look like from the phenotype, the outside of it. And we also look at their physiology. Do they function the same way as another organism that looks like them? You know, then we look at the biochemistry. How do they do? Do snakes have the same venom, or do they produce different? Those types of things we're looking at. Then we look at the DNA. We can take a DNA now with our supercomputers. We can take an organism and kick out its genome. We've done it with soybeans, corn, humans, all sorts of different animals. We're working on getting the genomes for these different species. So not every species has the same genome. There's a little differences between them. You know, monkeys and apes and humans are very, very close in our genetics, but not that close. I think like 4% difference is all in some of the apes compared to us. But a species is a group of populations, a group of individuals in a population that are the same. They produce viable, fertile offspring. So if you cross a horse with a donkey, you get a mule, right? They're not viable, so it's really, they're a kind of a subspecies. They're not really their own viable individual anymore. They can't reproduce with another mule to form a mule. They're sterile. So we keep that separate. Species are going to be separate from each other, and they can't interbreed to make a viable individual. Ligers, you know, they can breed a tiger and a lion together. You can breed different horses, different cows, but you can't do a horse and a cow. Too far spread in species. Now, at some point in evolutionary history, they were the same animal. They've just diverged out, you know, millions of years. They've changed to what they are now. But the gene flow will keep everything in place, make sure that we have a variety in our species, and it gives us diversity. You know, the big thing now is diversity. Everybody wants a diverse population, lots of differences, different colored skin, hair, religions. All these things are part of the diversity that we look at. You know, but eventually, if you look at us, we all are the same. We all have the same genetic beginnings, and we've just evolved. We've adapted to those differences. Any one of these here people that's on this picture right here could breed. They could mate with one another. They are still the same species. They just look different, right? So historically, we're all still the same species. We get to the point where we diverge. If we would have kept Australia separate for another million years, the Aborigines may not have been able to breed with us. Who knows? They may have completely mutated to the point where their offspring and our offspring mix would not be viable, be two different species. And who knows, you know, that might have happened to the Neanderthals and the Cro-Mags. We weren't there to see that. So, reproductive isolation is when we have barriers. Let's say we have a population, and one population goes hunting one day, one half the population goes hunting one day, and all of a sudden there's a big volcano, and a mountain range spreads up between the two. It's kind of highly unlikely, right? But it could happen in SimCity or something. So we know it'd be called Sim Mountains or something. So they have this mountain range. Now they can't get over the top of them, so now these two populations are going to diverge, or they're going to become their own populations because of mutations and reproduction and independent assortment, and all these different things are going to take place. So now they're going to be two separate species eventually, or so separate that they cannot change. They cannot interbreed. Hybrids are offspring that crosses between different species. So a liger, lion and a tiger, that would be the ones that we look at.
What about the pig-human thing they just made?
Pig-human thing? I don't know if
It's some chimera that they supposedly made in the lab.
They've been using pig valves. they use pig valves in people's hearts,
Yeah,
so,
they're wanting to grow human organs instead of pig or something like that.
yeah, I don't know. We had a nice discussion the other day about cloning and stuff like that, and I guess we never really talked about using other organisms, but most of the time there's so much difference between them that we can't use them. The different species doesn't allow for our immune system to accept them. But I know they've been doing a lot of genetic modifications on pigs. So I even saw one on YouTube or somewhere that was painting. It picks up a paintbrush and makes paintings. So they say they're very smart. I don't know. Pretty tasty sometimes too. Especially if you make bacon out of them. So I don't know. I don't know that one yet. So we'll see how that turns out. Okay, so prezygotic barriers block fertilization. So prezygotic, they can't even attempt to mate. They can't get together. This would be like a chastity belt, basically. You keep people from having intercourse or keep our organisms completely separate until the point where they no longer can interbreed again. So changes take place. Habitat isolation, again, we talked about that. Even though it's not completely isolated by barriers, they just kind of stay by themselves. They say, okay, you live on this side of the on this side of the island, I'll live on this side of the island, and we are okay. Or our niche is over here, we find food better here, you find food better over there, and they kind of separate and change. Even though they could possibly get together, if the wind was right, or if they wanted to walk over there or fly over there, they could breed, but they stay separate just because of choice, basically. So temporal isolation or time isolation basically is if you have two organisms that come in heat or estrus at different times, they cannot breed either. So, you know, farmers have been playing with this for years. When do you bring your, they have special times. The old farmers will tell you, you bring your bull in at one time and you put the buck sheep out at another time, and vice versa. They know the gestation periods and when the females will come into estrus or heat, and then they can release the male at that time. They keep them separate so that they can dictate when their babies come. Now, most of the farmers like to have their sheep come in about this time of year, now in February, so that they're ready for spring. They have a better price around Easter, and the other farmers that want them for grazing can buy them, and they do pretty good. But if they wait, so they time it so that 6 months from before that, they let the buck sheep out, so he can breed with all the females, and then the babies will come at that time. Now, that doesn't always work, because if they don't come into heat at that time, then they can't get pregnant anyway. So they've kind of manipulated it over the years so that they can work with that. Otherwise, you'd have babies all year round, and they can manipulate that. Especially even with humans. How many days out of the month can a female human get pregnant? Something everybody should know. What do you think, guys? How often can you get how many days of the month can you get a girl pregnant? You got 30 days. Give me some numbers. What do you think?
4.
4?
Who knows?
You're really close, man. Anybody else? 5 or 6? You're going to spread out just a little bit, right? Hedge your bet. In actuality, on a normal basis, 3 days max, about 72 hours, is all that a female should get pregnant. Because you have 1 day for ovulation, the egg and the sperm are only viable for about 24 to 36 hours, either way. So That's about it. 3 days max. The egg is only good for 24 hours. The sperm is only good for about 48 hours. So you have this window of about 2 days to 3 days max that a female should get pregnant. Now, that's right around the time of the ovulation. If everything goes right, and if she's ovulating on the right day that she normally should ovulate, right? But sometimes stress, poor diet, no sleep can make a female ovulate at a different time of the month. So there are possibilities of getting pregnant other times if her ovulation cycle is off. See, even during the menstrual flow, she can get pregnant if there's a problem with ovulation in the ovaries. So there should be normally, usually 3 days maximum in the month that she should get pregnant. There's a lot more to it than hopping in the old Ford and getting pregnant. Right? It's quite the process, but you have to be in the right situation. And a female, when they are female human, when they are ovulating, they are more receptive at that time also. So just like any other animal, when a female deer comes into rut, they are more receptive and they give off that pheromones, those smells that attract the males even more. So it's inbred into us. We can't stop it. We don't even know we're doing it. We don't know that the pheromones are even there, but we know we detect them, and they are very important for us as far as our reproductive mechanisms. That's kind of what drives the desire of reproduction. If it wasn't for the hormones, that type of a mechanism, the subconscious mechanism, there probably wouldn't be sex in the world, because that's what drives it. Behavioral isolation, courtship rituals, other behaviors that make it so that we have effective barriers. Maybe they don't like each other. You know, you're ugly peacock, I don't like you, I'm not going to breed with you, and pretty soon they don't work. So if the behavior isn't right and the female doesn't select that male, she will go beyond her reproductive time and they lose out on that reproductive window. Mechanical isolation. Something happens, some type of morphological differences happen to prevent them from mating. At one point in our history as humans, the male genitalia was different than it is now. It was not as, I guess you call it robust, so much as it is now because of the differences in the way that we reproduce now. If you look at the male monkey or gorilla, their genitalia is not very long and they have to reproduce in a special way, in a certain way. We have kind of morphologically changed to the point where we can reproduce in different, or we can have intercourse in different mechanisms, different ways. And, gives us a little bit of variation, a little bit of gives us an ability to reproduce in ways that maybe not normal in our past. Gametic isolation. Gametes, one sperm from one species cannot fertilize another species' eggs. So even though that they can reproduce, they can have intercourse, they just cannot, the eggs just don't work. Too much differences in the cell markings. For instance, even in humans, there are instances where young males, when they start into puberty, they start producing sperm. The body doesn't recognize those sperm as self, and they'll attack the sperm and basically sterilize that young man. They kill all the sperm before they can be released, and that's a type of sterilization and gametic isolation. If you would deposit those sperm in a female and she responds to it, kills all the sperm, then reproduction doesn't take place either. The act of it does, but the continuation of the species was not produced. So the post-zygotic barriers prevent the hybrid zygote from developing into a fertile individual. We talked about this before. We can have reduced hybrid viability, reduced fertility, or we can have the hybrid break down. It just doesn't work right. Another example of this basically is kind of, well, a mutation that's not very viable is Down syndrome. Usually they're not able to reproduce, and that's really not a product of zygotes. It's more of a product of a genetic separation, where the trisomy 21 creates that problem. So reduced hybrid viability, basically they just can't reproduce like a mule. It's not able to do so. This is a hybrid of two different, like salamanders, and this one won't be able to reproduce because of a hybrid. Now they were able to reproduce, but there's not going to carry on its traits. Another one is still sterile, reduced hybrid fertility. Sometimes they can't. So the donkey and the mule, donkey and the horse comes together, we get a mule. Now mules are really, they're bred for in certain cases. I know an Amish man, he loves mules. He would rather have a mule to work his fields than horses. They're stronger, they're more stable as far as working. They can work longer. They have better wind, and but they're about as dumb as a box of rocks. He told me once he had a pair of them that every time they would come over this hill, they would recognize that they were going home and they would take off at a dead run. He says, I could not stop them. He says, I tried everything. I put wire in their mouth to pull on. I put barbed wire in their mouth to pull on. They would keep going. They could not stop them. And they'd dump the wagons and everything because they would just take off running like ghosts. And then sometimes he says, I'd go like into somebody's farm and there'd be a bird on top of a wire. And those mules would see that bird and they would not go underneath that wire. They would just have these fear in them that he could not make them go across. So not only are they sterile, but they're not really that bright as far as what they can be trained. But they'll pull all day long. So someday maybe when you'll have a mule and understand all that. So some of the first generation hybrids are fertile, but when they mate with another species or with either parent species, the offspring are just weak and they're sterile. They don't make it. You know, you just can't. They seem very aggressive and very gregarious, or they're really strong, but when you breed them back again, it weakens that species. And you'll see this even with hybrids, with individuals that are very purebred. You breed two dogs together and it seems like their traits eventually get weaker and weaker. They get things like hip dysplasia and poor eyes and kidney problems and things like this because they've been inbred so many times that it creates problems. So not only do you, by breeding hybrids together, but by breeding the homogeneous genetics together can cause problems too. But usually when we breed individuals that are mixed, kind of a mutt, they come out with a stronger individual. You take two dogs from the kennel and you mix them together, a lot of times they're a stronger dog than you have if it was a purebred, right? How many have dogs in here? What do you got for dogs? What do you have?
I got a mutt.
A mutt? A big butt? Little butt?
Little butt. It's
Little
like
mutt?
a Jack Russell Terrier size.
Jack Russell Terrier. Do you know what's in it?
We had a test done last year, but it's a whole array. He's an alphabet soup.
57s? Yeah. Anybody else? What else you got? What do you got?
I had 2 Chihuahuas.
2 Chihuahuas? big guy like you and two chihuahuas. That's that's unique. What's their names? Spike?
Kevin and Roxy. I think they're the biggest dogs in the world.
Oh yeah, they strut it, don't they? What else? What do you got?
Labs.
Labs? They're all over the place. Labs like to, let's go, come on, let's go, come
I've
on.
had Cocker Spaniels in the past.
Yeah.
I've had a couple months that lasted forever.
Yeah. What's the oldest dog you've got? You had?
I had one that was 16.
16? Chihuahua?
No,
No.
he was a mutt.
Anybody have a purebred that lived a long time?
We had an Eskimo that lived like 12 years, 13 years.
That's pretty good for those.
Yeah.
Most of the time, the mutts do live better. They have better genetic variation so that they have a wide variety of traits that keep them alive longer. And that's not always true, but a lot of times that's true. Whereas the hybrids have more inbred diseases and problems that they can't get rid of. So, but dogs are cool. Dogs are probably the neatest things you can get. And they keep us grounded, I think. So it's good. So rice plants and corn plants and all these plants have to be bred. We breed all these organisms to suit our fancy. Look at the corn plants we have. They're huge. The rice plants produce more rice than ever. Wheat plants. You know, you go out west, there's just fields and fields of wheat. Millions of acres of wheat that now they're hybrids. They're producing more than they ever did in the past. So are they going to be viable? Or are they just a seed now? Sometimes these plants, you take two organisms, batch them together, plant the seed, and it grows, it produces lots of seeds, but it can't reproduce very well at all. The plants are weaker than the first generation of the hybrid that they produce. So we still keep trying to make a super plant that we can reproduce over and over and over again. But nature is probably the best at this over time because it allows for adaptation to the environment and what's going on with things. So we can do a certain amount of things and change, but we're only here for a little while. So species, biological species concept cannot be applied to fossils or asexual organisms. We don't know how they bred. We don't know, and prokaryotes, they interbreed by themselves. I mean, they're just binary fission. They separate, you get mitosis, and you have a really fast reproductive system. All right, I've talked past my hour, so you want to take a little break? We're going to do a little bit of a lab today. One of the problems with having lab twice a week is that it's really tough to put a big lab together unless we do one at a time. So we're going to talk a little bit about descent of species and how this all comes together, and that's about it for today.
It was fun.
Is that the word? I mean, it wasn't like ridiculously hard, but I couldn't, for the last few problems, I couldn't like look at it and figure out how to break it down. And doing Henry's Law, did you have to look up on Yahoo how to do that? Because the one in the PowerPoint isn't the one on Yahoo.
What'd you say?
Yahoo had like K equals P. No, it was P equals KC or something. K over C.
For which one?
For the Henry's Law one, where you have to figure out Henry's constant for
Oh.
whatever Kirk was going on?
No, I had to go like 3 or 4 different ones before I finally found out the right one.
Yeah.
They give me the runaround again, I swear to goodness.
Captain.
They keep not giving me my transcript. Yeah, I'm trying to get a copy of my unofficial transcripts. Okay.
Yeah.
Yeah. It's been like 3 weeks now, and like he said, the only reason why I accepted that you by now is I'm waiting on the transfer.
Yeah.
First class.
I had an issue with getting,
I don't think it.
so when I had to apply to Drake, my Kirkwood one sent easily, which is weird, because that's the one that has my maiden name on it. But this one from the ICC took forever, and I had to send it myself. It was copied and pasted it from whatever, like eBridge or whatever, and just sent it, to prepare this.
Yeah, well, they keep, I'll just go online and sign in. Or just go online and
It's been years, people.
Well, I don't, my username is not good anymore. I tried.
Yeah.
And they said, well, go online, I can do this. There are other numbers, guess phone numbers. And then,
I just, everyone I know who's gone to California has been like,
I hated people that,
yeah.
it was horrible. And they, like, cost me $6,000 for no reason. Their stuff is worthless, most of their classes will transfer.
I'm trying to think of what that, is that the one, is that in Cedar Rapids?
There's one in Davenport, that's one I went to. Kimberly.
I'm trying to remember what the school is up in Cedar Rapids that my friend's mom went to for nursing or whatever, and then she graduated and she tried to get a job and no one wanted her. No, it was in Kirkwood.
You said that pretty perfect.
So what do you recommend for students who are terrified of getting in front of a group and trying to teach? I'm sure you've had one who's,
Just realize that they're going to do the same thing.
yeah.
And that thing, what,
Small group.
picturing them all,
Small group.
yeah, just picturing them all naked doesn't work. That's not going to work. So, no, it's something that I think everybody needs to do is to get used to speaking to groups. If you're going to be in healthcare or even like some of these guys are going to be in conservation, you're going to be a DNR person, they're going to bring classrooms to you full of kids that you're going to have to talk to. You know, what is your major?
Biochem.
Biochem. Well, if you're going to be in the lab, you probably won't be doing a lot of speaking, but
Yeah, I figured that was the best place for me.
if you're going to go out though and do maybe be a chemical salesman or something, that's something you're going to have to talk to a lot of people about. I had a friend of mine, he worked for a company, and that's all he did is he did presentations to farmers. He'd buy them supper, bring them in, try to get a Ciba-Geigy, try to get them to buy this product. He made a really good living at it. I mean, that was the best he ever made. But he was gone all the time, and he just talked and talked and talked. Maybe that was what he liked to do. So there's a lot of jobs out there where you need to be able to function in front of people. I don't get nervous in front of students, but if I have to talk to my peers, I get shaky, I get nervous, still, after all these years. So if I have to talk in front of the faculty, it's completely different than talking in front of students. I've done this for so many years, it doesn't bother me.
Yeah, but these are my peers, so.
Well, just imagine that they're not. You know,
Okay.
it'll be a good experience. When it's all done, you'll say, oh, that wasn't so bad.
Yeah, I'm slowly getting my feet wet because I'm a Girl Scout leader, so like I lead and I teach in that, but they're all
Good.
like second graders, so.
This should help you with
Not
your
hard.
confidence and all that stuff. So between this and the article reviews, you're going to be an oratory before you know it. You'll be just like Abraham Lincoln. You'll be able to stand up and say anything you want. All right, so a couple things. We've got some two little videos we're going to do. One is on hybrids, some new hybrids. Actually, we were talking about pigs and humans, so I've got like a 1 or 2 minute one for that. And then I've got one on where did corn come from. So kind of goes along with this, is changing corn, making hybrids out of it. And so let's see what we can come up with this one here. Oh, wait a minute. I think we're muted. Here we go. Now we'll start over again.
All that creepy pigments.
I don't know if they talk or not. Okay, so let me explain this. What's going on? What are they doing?
Making works.
Making orcs? Oh no. It's called Gandalf. So you got something going on here, right? So, how's this going to work? Why would they do this?
Is the hope that people who need those organs would be able to accept them easier if they were from a pig rather than from a human body? Because it brought up that often those transplants don't work. They get rejected
Right. At one time, years ago, they were actually tried to use baboon hearts in humans. They thought, well, they're baboons, they're close to humans, we'll just take a baboon heart, put it in, until we can find a new heart and transplant it. So you're going to use it maybe not as a permanent heart, but as kind of a stopgap in between. Failed miserably. They just rejected them wholeheartedly. Poor pun, sorry. But it was not a good thing. So they went to the Jarvis artificial pump, and to carry this big tube comes out of them, and it was just a mess. So now they're trying to do this with pigs, where they can make kind of an inert tissue that the body won't respond to or won't react against as readily as they would, say, another human heart or something from another species. Species to species, the same species sometimes works, sometimes it doesn't, but from different species is very difficult. They have different markings on the outside of the cells, and the bodies will attack those organs very readily, kill them. So by doing this, hopefully, they can change the markings on the cells of the pigs so that they don't react to them as much. They've done this with heart valves. They can do that with heart valves now. They put in heart valves and they work pretty good. They don't react to them. You don't have to take medication with them at all. So I don't know. This is really the first I've seen them, what they're doing with it. So maybe it'll come out okay in another 20 years, like they said. Comments? You think that'll work? I don't think we're right to the point yet where we can just put a couple of, cells in a soup, some type of a goo, and make it grow. We have to have some type of an oxygen supply, a nutrient supply from some type of animal. So that's what they're doing. They're using that animal's mechanisms to grow those organs. Now, is that ethical? Is that right? I don't know. That's another question. You're going to raise this pig up, kill it, and what do you do? The rest? Eat the rest of it?
Isn't it part human?
Yeah. Thanks, pig. That's cannibalism. I just made it kill a part human. Just killed an orc and ate it. All right. So that's one thing. Let's take a look at this one for the, let's see, Pop Secret. This is from the Howard Hughes Medical Institute.
$3 for 6.
Okay,
All right, thank you.
great, thank you so much, have a great day. This is an everyday scene, but it's actually pretty amazing. We've taken dozens of wild plants and transformed them into useful crops through the process of domestication. Humans have carefully bred these plants for generations to make them bigger, sweeter, more colorful, and it's hard to find a plant that we've transformed more completely than this one, maize. Here in the US, most of us call it corn, and we eat a lot of it. There's cornbread, corn chips, corn cereal. If you look a little deeper, you'll find cornstarch and corn syrup in hundreds of products. And a lot of the meat we eat comes from animals fed a corn-based diet. So maize is all around us, but for a long time, the origin of maize was a mystery. The ancestors of wheat pretty much look like wheat. The precursors of apples basically look like apples. But there's nothing in nature today that looks like this. This is the story of an unexpected collaboration. The story of geneticists and archaeologists working together to discover where maize really came from. Christopher Columbus's crew were the first Europeans to see maize. But by the time Columbus arrived, people all over the Americas had been growing maize for thousands of years. Archaeological evidence from around the world reveals that starting around 10,000 years ago,
humans were beginning to live in larger settlements and to manipulate wild plant and animal species to better suit their needs. In the case of plants, this process of domestication led to plants that we call crops, like wheat, apples, and potatoes. And in most cases, the wild relatives of these crops can still be found in nature. But you can't find anything that looks like maize growing in the wild today.
And even the earliest fossil ears of maize, which are more than 6,000 years old, already look essentially like today's crop. So where did maize come from?
Many scientists thought that the ancestor of maize must be extinct. But a brilliant young geneticist discovered something that made him think that the ancestor of maize was right in front of us. His name was George Beadle. Beadle was studying a grass from Central America called teosinte. He found that teosinte's chromosomes looked nearly identical to those of maize. He also showed that teosinte and maize could produce fertile hybrid offspring, meaning that they must be closely related. Beadle concluded that teosinte was likely the ancestor of maize. But many botanists doubted the young scientist's claims. Maize expert Dr. John Dobley at the University of Wisconsin told me why.
So Neil, the reason I wanted to bring it out here is to show you just how different corn and teosinte are.
Yeah.
This is a teosinte plant, and it doesn't look anything like a typical corn plant.
No.
And you can start by just looking down at the base. It just branches a lot, so it is a very bushy creature, and quite different from a corn plant, such as you see here,
Yeah.
where there's just a single main stalk, no branches, except for these two short branches, each of which has an ear on it.
The dramatic difference in branching between teosinte and maize is just the beginning. When you look at an ear of corn, you can see hundreds of kernels exposed on the cob. But teosinte is different. Each ear only has a handful of kernels, each enclosed in a fruit case that's so hard you might crack a tooth if you tried to eat it. It was no wonder that botanists doubted that teosinte could be the ancestor of maize. Beadle moved on to other questions in genetics, which ultimately earned him the Nobel Prize. But the origin of maize continued to intrigue him. And after his retirement, he returned to that question.
To silence the skeptics, Beadle had to show how humans could have transformed this into this. So after his retirement, he launched one of the biggest breeding experiments in history to settle that question once and for all. For Beadle, the key question was how many genes control the differences between maize and teosinte. If that number were small, then it wouldn't have been too hard for early humans to transform teosinte into maize.
He began by crossbreeding maize with teosinte. In most plants and animals, individuals inherit 2 copies of each gene, one from each parent. So the offspring from this first generation cross between teosinte and maize, the F1 generation.
Okay, how do they do this? How would you do this in the wild, in your field, if you're a farmer? How could you do this? Cross-pollination. How do they do that? Anybody detassel corn when you were a kid? Do you know how that works? Why do they detassel corn? So if you have one plant, you have the prehistoric plant and our modern plants, and you grow those side by side, they're going to cross-pollinate, correct? Because they both are angiosperms and they have the capability of producing pollen and receiving pollen, right? Everybody, we'll talk more about that as we go through the semester. But when you detassel corn, you put the corn in rows. You put 2 rows or 3 rows of corn that you want to plant, so there'll be female rows. And you have 1 row of male plants that has the pollen in them. Those rows that you want to have as female plants, you take their tassel off. You get rid of their tassel so that the pollen does not come down onto the silk and pollinate that plant, or they don't cross-pollinate themselves. So the pollen from the male row that has the traits that you want put into the female plants will cross-pollinate over and settle in on them. You'll get the traits from the male into the female and reproduce that way. You see? So by taking the tassels off the females, putting the pollen from the males onto the females, you now have transferred those traits. That's what they're doing here. Now they're probably doing this in a greenhouse, but that's kind of what the farmers would have done in those years, right? So at some point they had to have some type of cross-pollination, some type of mutation so they could cross-pollinate back, and they kept making it better and better plants as it went along.
Would have one copy of each gene from teosinte and one from maize. These F1 plants would then be crossed with one another to produce the F2 generation. This is where things get interesting. If only one gene differs between teosinte and maize, then 1 in 4 of the F2 plants should look just like maize, and 1 in 4 ought to look like teosinte. If 2 genes are at work, this number drops to 1 in 16. For 3 genes, it's 1 in 64, and so on.
If more than 3 genes were involved, Beadle is going to need a lot of plants. He decided to grow 50,000 F2 plants for his experiment.
And what did he find? About 1 in 500 plants looked identical to teosinte, and a similar number looked just like maize. That number suggested that changes in just 4 or 5 genes were responsible for all the major differences between the 2 plants.
So George Beadle was right. The real ancestor of maize was teosinte, and it was right in front of us all along.
Many varieties of teosinte grow throughout Mexico and Central America, and humans have lived there for thousands of years. So where and when did they first transform teosinte into maize? Dobley's team set out to find the answer. They collected DNA samples from different teosinte varieties throughout Mexico to compare their DNA sequences to those of modern maize. The more closely related 2 groups of organisms are, the more similar their DNA sequences will be. Dobley's team looks for the teosinte variety with DNA sequences most similar to maize.
We've actually figured out that all of modern corn traces back to one type of teosinte in the southwestern part of Mexico, near a river called the Balsas River.
The relatively small number of DNA sequence differences between maize and the Balsas River teosinte yielded another critical piece of information.
We can take teosinte in corn and ask how many mutations do they differ by, and then knowing the rate at which mutations occur, make a prediction about how long ago their paths separated.
The more differences in the DNA of 2 groups of organisms, the longer it's been since their ancestors were all one species.
Our estimate is that the original domestication of corn would have taken place sometime around 9,000 years ago.
Based on genetics, Dobley's team had come up with a hypothesis about where and when maize was domesticated. But the ultimate test would require independent evidence from outside the field of genetics. I visited Dr. Dolores Paperna at the Smithsonian Tropical Research Institute in Panama to see that evidence.
So you're an archaeologist. What did you think when this geneticist from Wisconsin analyzing DNA said, here's where we need to look for the earliest evidence of maize domestication?
Teosinte is distributed all over Mexico, highlands, lowlands, it gets down into Nicaragua. So the question for archaeologists was, where do we go? And Dr. Dobley's work told us exactly where to go.
9,000 years ago, people living in this area were taking shelter and preparing food in caves and rock shelters.
When we went to the Central Balsas Valley, one of the things we did is to ask local people, do you know of any caves or rock shelters? And that's how we found the Xihuatoshta shelter.
So people took shelter there, they slept there, they probably ate there.
They ate there, they cooked their food there.
But finding evidence of ancient maize wouldn't be easy. In the tropical environment of ancient Mexico, the cobs and kernels would typically be scavenged or decomposed. But Dr. Paperno wasn't looking for such obvious evidence.
These were the earliest plant processing tools. We call them plant grinding stones, that's what they were used for, and these are no more than river cobbles.
Dr. Paperno showed me how ancient people used these stone tools to grind up maize and other crops. In the process, tiny plant pieces might be deposited on the tool's surface, leaving behind microfossils.
So we found hundreds of these microfossils right on the grind surface of the stone. And like the seeds, they're very highly diagnostic.
So even with these microscopic traces, you can tell the difference between corn and teosinte.
Yes, we can tell the difference.
Finding maize microfossils on the grinding tools meant that the humans living in the Xihuatoxtla shelter were processing maize for food. But how long ago? Archaeologists can calculate the age of ancient remains using radiocarbon dating, but microfossils are too small to date using this method. So Dr. Paperno used charcoal found in the same sediment layer as the grinding stones to determine the age of the microfossils.
And so what was the oldest date of these maize remains?
It's very interesting how well the genetic and archaeological data fit together. The oldest charcoal date we received back was about 8,700 years ago.
That date coincided almost perfectly with the date Dr. Dobley predicted from the genetic evidence. So nearly 9,000 years ago, humans had already produced an early version of maize. But how was teosinte transformed into maize? Back in Dr. Dobley's lab in Wisconsin, I learned about the genetic changes involved.
One of the main differences between teosinte and maize is that the teosinte seeds are encased in this really hard fruit case that makes it really difficult to eat. So clearly, that's something that had to change.
That's right, and the remarkable thing is that having a fruit case versus not having a fruit case is basically controlled by a single gene.
A single gene.
A single gene.
To test this gene's function, Dr. Dobley's team did a clever experiment. They carefully crossbred maize and teosinte to introduce the maize version of the fruitcase gene into teosinte plants. When they did that, the teosinte kernels, which are normally enclosed in a hard fruitcase, became partially exposed, almost like little corn kernels. When they did the opposite, putting the teosinte fruitcase gene into maize plants, the fruitcase became larger and started to cover up the maize kernels, similar to teosinte.
One gene makes a pretty dramatic change.
So another really obvious difference between teosinte and corn is that teosinte produces dozens of these little tiny ears on a plant that branches a lot, and corn just produces a couple of ears on a plant that hardly branches at all. So what's going on there?
There is one gene that we've identified that plays a central role in that process, and you call it the branching gene.
Dr. Dobley explained how putting the teosinte version of the branching gene into maize made the maize plants more branched, like teosinte. And putting the maize version of the gene into teosinte made the teosinte plants less branched. Dr. Dobley has shown that the fruitcase gene, the branching gene, and just a few others, a small number of genes, just as George Beadle predicted, were responsible for setting in motion all the major differences between maize and teosinte. But how could so few genes cause such huge changes? Why were these genes so powerful?
They both belong to a special class of genes called regulatory genes. And these are genes that directly regulate the activities of other genes.
And so when we move the teosinte version of one of these genes into a corn plant or vice versa, We're actually changing more than just that one gene.
That's right, they can turn other genes on and off. You can think of these genes as something like the conductor of an orchestra. And if you would take the conductor from one orchestra and give that orchestra, say, a new conductor,
Just like we did moving some genes from teosinte to
Right.
maize or vice versa.
and you could get a very different quality of music, even though all of the musicians and all the instruments remain the same.
These regulatory genes probably influence the activity of hundreds of other genes, which explains how mutations in just a few regulatory genes could dramatically transform teosinte. But there was still one thing I couldn't figure out.
So I understand now how teosinte was transformed into maize, but the thing that's still bothering me is that teosinte really doesn't seem like a very good crop. So why would anybody have started growing it in the first place?
Well, George Beadle actually had an idea about that question, and his idea was that they might have used it like popcorn. And Beadle did an experiment to test his hypothesis that they used it like popcorn, and we can do that same experiment here today.
All right, let's do it.
Remember, the nutritious kernels of teosinte are trapped inside hard fruit cases. But if they popped like maize kernels, that could be one way the earliest farmers could have eaten teosinte. In Dr. Dobley's lab, we were about to find out whether the ancestor of maize could pop.
Oh, that was a good one. Okay, so we've got some pop teosinte here. And I'm gonna actually give this a try.
It's good for me.
And that's basically just like popcorn.
It tastes like popcorn.
The archaeological and genetic evidence tell us a remarkable story. About 9,000 years ago, people living in the Balsas River region of Mexico began growing an unassuming grass called teosinte and ended up transforming it into the amazing crop we now call maize.
Okay, so what do you think? Is that something we should have done? You know, so we go back to always the question, you know, are we playing God when we're doing things like this, or are we just making it better so we can survive? What are those people, were they intending to change something for their own fun, or were they doing it for...
They were doing it for the benefit.
How do you suppose they got from popcorn to corn?
You realize you can use it for popcorn, so you start growing it more and more, and then you realize that you can breed it
Take the
together.
ones that look
Yeah.
better, keep them closer together back then, I guess. I don't know.
Well, and
They
maybe
wouldn't
if
have
they
it in the
grew,
lab.
they grew so much of it, remember like they said, 1 in every 500 plants,
You accidentally get a couple of those.
you get a couple of really corn, and then you plant those, and you plant those, you know, together, and they start growing whole fields like that. So, yeah, so now you have popcorn, and you have field corn or maize, like you call it. Now, is there a difference between sweet corn and field corn? What's the difference?
Not really sure the major differences, but I just know field corn is more starchy, and it grows more ears than sweet corn.
Can you
does.
Can you eat field corn?
I mean, you can, but it's not very good. It's not sweet.
Has anybody ever tried field corn?
Yeah.
Yeah.
If you eat it when it's just starting to grow an ear, it tastes like sweet corn, but later on it starts getting really starchy.
Bitter.
Yeah.
Or not. Dry.
So we got popcorn and we got yellow corn from 9,000 years ago. Where do we get sweet corn from? Is the sweet corn what they were growing 9,000 years ago, or was it the field corn?
Beautiful.
Probably still kind of it's almost like the primitive corn, the teosinte, and the regular corn that we have now. probably that hardness at the end when it dries out, that's what kind of get, because if you ever tried to chew a corn seed, corn kernel, they're really tough. Maybe not as tough as the teosinte, but pretty tough stuff, right? You break a tooth on them pretty easy, at least I do. My wife won't let me eat those anymore, I love them. But, so somewhere along the line, we modified to create this plant called sweet corn. Now, is sweet corn the same as what it used to be? Or have we changed it ourselves? Or where do you think we got the idea to do this?
I'm pretty sure we've changed it quite a bit, because aren't the crops that we have now a lot bigger than what they used to be?
They're taller, they're bigger, they're more, the ears are longer. But we still have that difference in texture, that difference in flavor, and the difference in hardness of the kernels. How do we get that? How do we get those things? Did we use the same process that we did going from teosinte to regular maize corn now? To make the sweet corn? How do you suppose they figured that out? Somebody just picked up an ear, hey, this is pretty sweet. This one tastes better than the others, what's going on? So maybe some of this was accident, some of it is genetically changed, we've bred them that way. You know, the farmers over years have modified and physically planted one row next to the other row, or one corn plant next to the other corn plant to get specific traits to make our nice little sweet corn. Now there's different types of sweet corn too. Right? You go on, some of the sweet corn is all yellow, some of them are the candy, or whatever, candy sweet corn. It's got
Peaches and cream.
Yeah, peaches and cream or whatever they call it. So, yeah, we've changed those things a lot, but they're all hybrids. They're hybrids of that teosinte. At some point, that was the beginning, and we branched out from that. So, when we look at what we're talking about in class and how the hybrids work, we've pretty much hybridized everything as far as corn from that teosinte plant. Then rehybridize it year after year after year till we get what we have now. Now we just do it genetically in the laboratory. Genetically changing willy-nilly whatever we want. Put genes in it. So, yeah, for 9,000 years we've been working on making the corn plant to what it was. And until advent of science came around where we could genetically change them in lab, we still did that. We still did them in the field, manipulated the corn plants to make hybrids that were stronger, better, and more usable even at that rate. So hybrids around a lot. We're all hybrids, basically. Even as humans, we're hybrids. So, anyway, the only other thing I was going to talk about is genetics, descent of man. We really haven't got to that yet. Well, I'll hand this out. You can bring it with you next time. We're going to get to the, how old the Earth is and all that stuff as we go through the next couple chapters. 24, 25 are deal mostly with that. But you can take those papers and kind of look them over. Kind of shows the beginning of it. And actually, I think some of these might be a little bit need to be updated. They've got some now. Hominids are 4.4 million years old. And so in the scope of things, what were man eating before corn came about? What did we eat back then? It's kind of weird to think about, but we only 9,000 years had corn. And how many years of corn do we produce now? Millions and millions and millions of them. Would they say that we export 50% of our corn plants out of the country? So it gets kind of scary when we start messing with trade deals. If we say that we can't trade, if other countries can't trade to us, what are we going to do if they don't let our foods come into them? Okay, so we'll talk about the descent of man and how we all fit together as far as our evolutionary processes from some type of hominid to Homo sapien, sapien, I guess Homo sapien, and to what we are now. We will, so I will get to that maybe next lab time. That's what I really like. I like that. That's anthropology stuff. That's pretty cool. Okay, I'll let you out today a little bit, a few minutes early, and we'll get some more labs. Hoping the weather turns nice here pretty soon so we can start doing some field trips. And probably one of the first ones we'll do is go out to Discovery Park and go through that facility. It may take more than an hour to do that, so if people have to come and go, that's okay, but we'll make it work the best we can. And then throughout the semester, another indoor one we can do is the Button History Museum out here. But I like to go down to the hatchery place first, because they give us a good background of what's going to happen and what we see out there. I guess we could do both ways. I have to coordinate that with the fishery people. So that when they're actually working on breeding fish, then we can be out there and watch them. Okay, if you have any questions, come see me. If you have any problems, notice a lot of you haven't started your post-test yet, so just to read the chapter and start working on those post-tests. They're all underneath on the left-hand side under Course Home. Click on the unit, and then it'll open up the questions that you need to work on. The quizzes, only about 25 questions, or some of them are even less than that.
So the first one was like 10.
Yeah, it's just really short things, so don't fret them. Basically, so you kind of understand what is in the reading material. Okay, I need a 3x5 card with your name, date, and 2 questions on it. And we'll see you on Wednesday. Thank you. Thanks. 67 cents at Menards, 3x5.
Oh, I know. I'm going to go pick some up tonight.
I saw them last night. I go, 67 cents. That's pretty good for 100 cards.
I did that a couple semesters ago. I seen that they were just cheap, so I bought a whole bag of
Yeah, they're good.
those post-it notes.
I like just use them for notes. I stick in my pocket and off I go.
Post grocery list, it goes in the pocket easy.
Yep.
Doesn't crumble up like a piece of paper.
The post-it notes are always sticky, so you got to either fold those in half or. So these are like kind of nice.
They wind up sticking out my books like crazy. Yeah, another book from last semester. It looks like a cheerleader's pom pom, just because of all
Yep.
of them around it. But
Oh, they work great for that. Yeah, that's right.
I just got to write your little notes on them. Like right here, I got 22, 23, 24.
Mm-hmm.
Look right here.